Good morning, everybody, or afternoon, depending on when you're getting this. So I'm going to try and go through DNA structure a little bit more in depth than what we've been doing on the online, whether it's the CK12 or the Gizmo, just to give you a better understanding of what's actually going on with the DNA structure. Because as I said, this is very important, and there is a lot of things that kind of build off of this topic. So understanding the structure of it and kind of what pairs with what and how everything looks is going to be essential for us to keep moving forward. So without further ado, DNA. Now, DNA stands for deoxyribonucleic acid. When we start talking about RNA, it's ribonucleic acid. There are a couple similarities, a couple differences between the two, which we'll get into in a little bit. But deoxyribonucleic acid. Now, nucleic acid, again, keep in mind that nucleic acids are one of our macromolecules. We have proteins, lipids, carbohydrates, and nucleic acids. DNA and RNA are our two nucleic acids that fall in line with that macromolecule. So it is a double helix. Kind of picture a spiral staircase, or picture one of those rope ladders that you would hang down over a boat. Yeah, I guess that's the best one. Over a boat, and then just twist it a little bit. You get that double helix type of shape. Made out of three parts. We have a deoxyribose, hence the name, a phosphate group, and then a nitrogenous base, which can either be adenine, thymine, cytosine, or guanine. And those are all abbreviated with A, T, G, and C. What you'll notice in a minute is going to be what's known as Charkov's rules, which is every time A pairs with T and C pairs with G. So if we were to look at it over here, in our DNA structure over here, let's see if I can move me. I can move me. In our DNA structure over here, we could say this is adenine, and this would be thymine. Every time A and T pair together, and then G and C. So we could say this is guanine, this is cytosine. So if we go through it again, adenine, thymine, and we'll say guanine, cytosine. I think I said it the same way up there. Guanine, cytosine, adenine, thymine, cytosine, guanine, guanine, cytosine. A and T, G and C. Now, there's a way to remember it. I thought of a way to remember it. If it doesn't work for you, think of a different way. But an acronym always works here. So what I always said is we're always going to go shopping at Gucci. So that store Gucci. At AT always go together, and Gucci, Gucci, I think it's spelled. So G, C, they always go together. It works. Could be better, but it works. So here's our structure of a nucleotide. So our nucleotides are the building blocks of our DNA molecule, just like, well, actually take a minute. Think of if you remember the building blocks of each of the macromolecules. Pause the video right now if you are trying to think of it, because I'm about to spoil it and give you all the answers. So we had our carbohydrates, which are building blocks for going to be our monosaccharides or our sugars. Our proteins, which are building blocks for the amino acids. Our our lipids are going to be our other one, and our building blocks for those are going to be our fatty acids, and our triglycerides are going to be the building blocks there. And now our nucleic acids, we have our DNA, RNA. The building blocks for those are going to be our nucleotides. Nucleotide is made up of three parts: phosphate group, that's sugar, and our nitrogenous base. And then all these nucleotides make up the DNA molecule. Oops, wrong direction. So DNA is just a bunch of monomers, pretty much a bunch of these nucleotides, which again has that sugar, the phosphate, and the base. Now, the sugar here is deoxyribose. In RNA, that is just a ribose sugar. That's one of the differences between the two. The dimer is a dinucleotide, so two of them together, di, two. And then polymer is the polynucleotide, which is many of them, which our DNA is a polynucleotide. So here's our deoxyribose molecule, and then our structures of adenine, guanine, cytosine, and thymine. Now, you'll notice, remember before we said A and T pair together, and G and C pair together. So just looking at these diagrams, why is it significant that adenine always pairs with thymine and guanine always pairs with cytosine? So again, pause the video, take a minute, think about that, and then I'm going to give you the answer in a moment. Okay, So hopefully what you came up with is that it keeps DNA its shape kind of structural throughout. So if adenine and guanine paired together, we would have sections of DNA that's going to be very wide and sections of DNA that is very narrow. So keeping it with, and we can just say 1, 2, 3 ring structure and 1, 2, 3 ring structure. Well, it's actually these two and those two, but same thing. Keeps it even throughout, so it keeps it nice and uniform looking structure. So again, double helix ladder. The uprights are the alternating sugars and phosphates. So up here, we're going to have our sugar, phosphate, sugar, phosphate, sugar, phosphate. That's what's known as the sugar phosphate backbone. Okay, and the sugar is always going to connect to that base. The base is going to be these rungs of the ladder in here. Notice it takes two bases because our DNA is a double helix. It's two-sided. We have two sides of DNA, the one side here, the other side here. So we need a nucleotide on this side that matches with the complementary nucleotide on the other. When I say complementary, all I mean is the nucleotide that matches with it, A with T, G with C. Again, the rungs are the bases, and we have hydrogen bonds that are going to hold the sugars to each other. Which is the middle of the ladder. Right here, these are our sugar bonds. Notice here we have a double bond compared to a triple, double, triple, triple, double. And then we have covalent bonds that hold the alternating sugars and phosphates together, the uprights. Now, that is going to be very important because think about what we know about bond types. Is hydrogen bond strong or weak? Hopefully you all said weak. It is a weaker bond. So when we're going through DNA replication, which we're going to talk about in a little bit, that DNA actually has to separate. So if our DNA is like this, it actually has to start to separate in order to copy the DNA and replicate it. So having those weak hydrogen bonds in the middle is very significant to allow it to do that easily. The backbone here, we don't want to break. We want that to stay steady. So we have covalent bonds, which are much, much stronger of a bond than those hydrogen bonds, so it doesn't break apart. Now, the four bases in DNA can also be classified as being purines or pyrimidines. And there is a way to remember these as well. It's a little more confusing than my shopping at Gucci, but we'll get through it. And again, if you think of anything better, feel free to let me know. So purine bases have the two rings, the adenine and the guanine. Those are both our purine bases, the two ring structures. So we have our adenine and guanine, two ring structures. Whereas the pyrimidine bases are one ring structures, are cytosine and thymine. Now, let's see. So let's go through this and I'll tell you the way to remember it. The base pairing rules. A pairs with T, C pairs with G. We've already talked about that. Hydrogen bonds hold these bases together. Adenine and thymine are held together by two bonds, are represented by the dashed line, and cytosine and guanine are held together by three bonds, which is the other dashed line. So if we go back to here. Now, when we're looking at it, the way to remember these, and this, again, could be confusing. What we're going to do, purine, okay, small word, right? Purine is much smaller than pyrimidine. However, the structure is bigger. Small word, bigger structure. Bigger word, smaller structure. Pyrimidine, smaller structure. Now, you're probably thinking, well, okay, that's cool and all, but how do I remember which ones actually are that? Meaning, how do I know that adenine and guanine are the purines? Okay, I know that they have two rings, but how do I know that it's adenine and guanine? Well, look at how the word is spelled. Pyrimidine, PY, cytosine, CY, thymine, THY, they don't have Y's up here. So your pyrimidines are going to be the words that have the Y's in them, and they're going to be a smaller structure. Whereas your purines have that bigger structure because it's the smaller word, and they don't have any Y's, so it has to be your adenine and guanine. Way to remember it. If it works for you, use it. If not, feel free to come up with something better. With that being said, that concludes the structure of DNA. So if you guys don't have any questions, then that is all. And thank you for watching. If you do have questions, please feel free to shoot me an email or in one of the assignments, just kind of make a question at the bottom. I'll be sure to get it and get back to you. But email is probably your best bet. Thank you guys for watching. Look forward to hearing from you.